Welcome to the Deep Dive. Ready to explore something incredible? Today, we're diving into the world of gene therapy. Buckle up because we're breaking down those microscopic delivery trucks known as viral vectors. By the end of this deep dive, you'll be able to drop some serious knowledge about how scientists are using modified viruses to tackle diseases. You'll be the life of the party. Let's start with the basics. What exactly is gene therapy, and why are we even talking about viral vectors?
Imagine this, treating diseases right at their source, the genes that make up our DNA. That's the potential of gene therapy. Replacing, removing, or adding genetic material to fix faulty genes. And the way we get there, viral vectors, the delivery vehicles for these therapeutic genes.
Okay, I'm with you so far, but, and maybe this is a silly question, why viruses? It seems counterintuitive to fight fire with fire, right? Using something that can cause illness to treat illness.
Great question. The viruses used in gene therapy aren't your average everyday viruses. They've been modified, see? Scientists have stripped away their harmful parts, turning them into harmless delivery trucks.
So it's like they're reprogrammed, turning these viruses into the good guys. Okay, that makes me feel better. Now, there are different types of viral vectors, right? And each one has its own quirks, its own strengths and weaknesses.
You got it. It's like picking the right tool for the job. And in our gene therapy toolkit, we've got four main players: Adenovirus, Lentivirus, AAV, and Retrovirus. Let's start with Adenovirus. Think of it as the workhorse.
A workhorse, huh? What makes adenovirus so special?
Well, it's a double-stranded DNA virus, which basically means it carries its genetic information in a specific way. But its claim to fame, its cargo space. Adenovirus can carry a surprising amount of genetic material. We're talking up to 36 kilobases.
36 kilobases? Whoa, is that a lot? To be honest, I have no clue how big a kilobase even is.
Fair enough. It's all relative, right? Think of it this way. Genes come in different sizes, and some of those therapeutic genes are quite large. Adenovirus, with its generous carrying capacity, can handle these bigger genes. That makes it super useful for therapies where you need to deliver a whole lot of genetic information.
Ah, so it's like choosing a bigger delivery truck when you've got a bigger package to deliver. Makes sense. But every system has its downsides. What about this workhorse? Are there any downsides to using it?
Always a trade-off in science. Adenovirus has the space, sure, but sometimes it triggers a strong immune response. Our bodies, they're smart, you see. They recognize it as a virus, even in its modified form, and try to fight it off.
So it's like our immune system throws a wrench in the works.
Precisely. This immune response can limit how long that therapeutic gene can actually do its job. And another thing, adenovirus, it doesn't usually integrate into our DNA.
It's more of a temporary visitor then.
Exactly. Delivers the gene, but doesn't stick around, doesn't become a permanent part of the cell's genetic makeup. Now, let's hop over to our next viral vector, lentivirus. This one's been making headlines.
Lentivirus. Now that sounds familiar. Is this the one involved in that CAR T-cell therapy I keep hearing about?
That's the one. Lentivirus is a type of retrovirus, and it's been instrumental in developing CAR T-cell therapy for cancer.
That's where they take a patient's own immune cells, tweak them genetically to fight cancer, and put them back in, right?
You're on top of it.
Yeah.
It's revolutionizing cancer treatment. And lentivirus, well, it's the star of the show. It has this unique ability to integrate its genetic material right into the host cell's DNA.
So, unlike our friend adenovirus, lentivirus becomes a permanent resident.
Exactly. And because of this integration, the therapeutic gene is expressed long-term, which is critical for something like sour T-cell therapy, where you need those modified immune cells to keep fighting the good fight, you know?
Okay, lentivirus is sounding pretty impressive. Sneaks in, makes those changes last. It's like a stealthy ninja. But there's got to be a catch, right? Every superhero has their weakness.
You're right. There's always a balance. Integration's good, mostly, but there's a slight risk lentivirus could integrate in the wrong spot in the DNA, could mess with how other genes work.
So like a typo in a sentence, but on a genetic level, could change the whole meaning.
Perfect analogy. Scientists are very aware of this and are constantly working on improving that accuracy. Now, ready for our next viral vector.
Hit me with it. We've got the workhorse, the stealthy ninja. Who's next on our viral vector team?
Next up, AAV, short for adeno-associated virus. Now, this one's cool. It's considered one of the safest viral vectors we have.
Safe is always a good thing, especially when we're talking about something as important as gene therapy. What makes AAV so safe?
AAV is a single-stranded DNA virus, and like lentivirus, it can integrate into the host genome. But here's the kicker. AAV tends to integrate at a very specific, very safe spot in the DNA, minimizes the risk of messing with other genes.
So fewer chances of those genetic typos we talked about.
Exactly. It's incredibly precise. Now, it doesn't have the cargo space of Adenovirus, but for treating certain genetic disorders, that safety profile makes it a winner.
It's like we were saying earlier, right? Picking the right tool for the job. You wouldn't use a hammer to tighten a screw. Each viral vector has its own strengths.
Absolutely. Which brings us to our final viral vector superstar, Retrovirus. Like Lentivirus, Retrovirus is a single-stranded RNA virus and can integrate into the host genome.
Okay, so another one with long-term potential. Where does retrovirus shine?
Retroviruses, they're the pioneers, some of the first viral vectors used in research. They've been studied extensively for a wide range of diseases. We're talking cancer, genetic disorders, even HIV/AIDS.
Wow, a true veteran in the field. But
Hmm.
my notes mention something called insertional mutagenesis. That doesn't sound great.
You're right, not the best. It can integrate into the host DNA, sure, but not always predictably.
So more of a random insertion?
You got it. And that randomness, well, it increases the risk of those disruptions we were talking about, interfering with how other genes function. It's a bit like throwing a dart at a dartboard, with your eyes closed, hoping for the best, but, well, there's a chance you might miss the target.
Yeah, a little scary when we're talking about DNA, but hey, at least scientists are aware of it, right? They're working on it.
Oh, absolutely. Refining those techniques all the time to make retrovirus safer and more effective. And that brings up a good point. With all these different viral vectors, how do scientists choose which one to use?
Yeah, it's like having all these tools, but you need to know which one to use for which job. It's a lot to consider, right? So how do they decide?
It's a bit like building a house. You wouldn't grab a screwdriver when what you really need is a hammer, right? Same idea here. Each viral vector has specific characteristics that make it a better fit for certain situations.
Okay, so walk me through it. What are some of those key factors researchers think about when choosing their vector?
Well, first things first, size. We already touched on this with adenovirus. Big cargo space, good for those larger therapeutic genes. But if you've got a smaller gene to deliver, AAV might be the better choice, especially with that impressive safety record of its.
Right, so size matters. What else?
They also have to consider what cells they're targeting. Not all viruses are created equal in that regard. Some are just naturally better at infecting specific types of cells.
So different viruses for different parts of the body?
You got it. For example, AAV really seems to have a knack for those cells in the eye. That's one of the reasons it's looking so promising for treating certain types of inherited blindness.
Wait, you can use a virus to deliver a gene that can restore sight. That's mind-blowing.
It's pretty incredible, right? And it just goes to show it's all about knowing your tools. Each viral vector has its own strengths.
Okay, so we've got size, cell type. What else is on the checklist when choosing a vector?
Another thing to keep in mind is how long you need that gene expression to last. If you're looking for a long-term effect, something like Lentivirus, which integrates into that host genome, that might be the way to go. But if you only need a short-term effect, a non-integrating vector like Adenovirus could be a better fit.
Fascinating stuff. It's like putting together a puzzle, finding that perfect match for each situation. So what about the future? What's next for these gene therapy all-stars? Anything exciting on the horizon?
Oh, absolutely. The field is constantly evolving, always moving. Scientists, they never stop. They're always working on creating new viral vectors and improving the ones we already have.
What kind of improvements are we talking about here? Give me the exciting details.
Well, for starters, they're trying to make these vectors even safer, minimizing the chances of those unwanted immune responses we talked about earlier. Remember how adenovirus can sometimes set off those alarm bells in our immune system? Researchers are looking for ways to make it less, well, visible, I guess you could say.
Stealth mode. I love it. What else?
Another big focus is precision. Improving the accuracy of gene targeting. We're talking about developing vectors that can deliver those genes to extremely specific cell types with incredible accuracy. Less risk of those off-target effects, where the gene ends up in the wrong place, potentially causing more problems.
Sound like gene therapy is only going to get more and more sophisticated.
Well, without a doubt. And as the technology keeps advancing, we're going to see even more innovative therapies emerge. It's a really exciting time to be following this field.
It's really incredible how far things have come, especially in such a short amount of time. From those first experiments with retrovirus to the incredibly targeted therapies being developed today. I mean, gene therapy has the potential to completely change how we approach medicine.
I couldn't agree more. It's remarkable.
My mind is officially blown. Who knew viruses could be so fascinating? To think we could use them like this to potentially cure diseases. It's like something out of science fiction. It really is amazing, isn't it? The creativity of these scientists taking these tiny things, these viruses, and turning them into something that could actually help people.
Absolutely.
Yeah.
But like with any powerful tool, we have to use it carefully,
Right.
responsibly.
Exactly. And with all this talk about, modifying genes, developing these powerful new therapies, well, it makes you think about the ethics of it all too, doesn't it?
It does. Gene therapy is moving so fast from the lab to the real world, and we need to be having those conversations, about access, affordability, what kind of impact it might have on future generations. There are no easy answers, that's for sure.
You're right. It's bigger than just the science, isn't it? It's about the human impact too. But on a lighter note, I gotta say, my mind is still spinning from all this. I never thought I'd find viruses so fascinating.
Glad to hear it. It's definitely a field that keeps you on your toes, that's for sure.
Well, thank you again for joining us on this deep dive and for sharing your expertise with us. I think our listeners learned a lot today. And to everyone listening, thank you for being here. We hope you've enjoyed this deep dive into the world of gene therapy and those amazing viral vectors. It's incredible stuff. And who knows what the future holds? What breakthroughs are just around the corner? Until next time, keep asking questions and keep exploring the amazing world of science.